Hello and welcome, AP Bio, and welcome to our Chapter 7 discussion on cellular respiration. Let's start off with a little misconception. Now, I'm sure you've heard your whole life, mitochondria is the powerhouse of the cell. Nothing could be further from the truth. In fact, mitochondria are the powerhouse of the cell. Mitochondria is a plural, so a mitochondrion is the powerhouse of the cell. These little guys, organelles, that used to be their own living cell, these used to be bacteria that our ancestors ate and instead of digesting, kept inside of their bodies to use as a resource to make fuel because they were a whole lot better at making ATP than our ancestors were. And so now this happy coexistence exists, this happy mutualism where we protect the mitochondria and the mitochondria provide us with energy. So, as we mentioned last time and talking about laws of thermodynamics, energy can't just be made out of nowhere, but it can be transferred. So when we talk about respiration pathways, we're going to be talking about going from complex organic molecules like glucose into ATP, which is our currency of energy. Now, this happens through a series of reactions that are controlled by enzymes, and some of them may require oxygen and be aerobic. Some may be anaerobic and not require any oxygen at all. But no matter which pathway is being taken, they're all going to start the same way. In fact, every single cell on the planet starts breaking down sugar with glycolysis. So, if we were to just break apart sugar to use it, you would have an explosive combustion release of energy that we couldn't really harness very effectively, and most of that energy would just be totally lost. But by doing a series of steps and a series of reactions, we can control that release of energy in that sugar and make a lot of ATP. So you may ask, well, why don't we just use sugar for our energy if that's what it's starting with? Why do we turn one form of energy into another form of energy to then use? Well, getting energy out of sugar is pretty complex. As I said, it's multiple steps in a really controlled release. That's not going to be as instantaneous as just taking a phosphate group off of an ATP molecule. So it's much harder to control for just that simple, get that energy. So then why do we even bother with sugar? Well, ATP is a really bulky molecule with just one little part that comes on and off for releasing energy. And so it's way too bulky to store enough to power our body through a day. So we are going to instead store sugar and then turn that sugar into ATP when we need it, because it turns out glucose has a ton of energy packed into it for a really efficient size. So instead of sending 40 ATP through the blood, send one sugar, and then once it gets to its destination, turn it into 40 ATP. So we'll break down this process on how this happens. So what are we going to see? Our good old friend C6H12O6, or glucose, plus a little bit of oxygen, and we get some carbon dioxide, some water, and some energy. So I want you thinking, is this reaction going to be anabolic, building, or catabolic, breaking apart? Is this reaction going to be endogonic, we're putting energy into the compounds, or exergonic, we're releasing energy? Well, we are going to be doing a catabolic reaction where sugar gets broken down because it's getting broken into smaller component pieces, and it's going to be exergonic because we're releasing energy. You may say, wait, aren't we putting energy into ATP? True. That is more of a separate reaction we'll talk about, but the initial just breaking apart of sugar is going to be releasing energy. So you may notice that I have reduced and oxidized here. What do those mean? Well, let's talk about redox reactions or reduction-oxidation reactions. These are chemical reactions where electrons are transferred from one atom to another. anytime you lose an electron or become more positive, because remember, if you pull away an electron, which is negative, you'll become positive by comparison, you're getting oxidized. If you gain an electron, you're becoming more negative, you're reducing because your charge is getting reduced by going negative. Now, how am I possibly ever going to remember that? Leo goes grr. So Leo the lion roars with a loud grr. What do these words mean? Well, if you Leo or lose electrons, it's oxidation. Grr, if you gain electrons, reduction. So if you can remember Leo goes grr, you will always remember what's oxidation and what's reduction. And as we're oxidizing and reducing all these molecules as we go through the steps, I want you to know about coenzymes. So I talked last lecture about coenzymes, how they're those helper molecules that help out by shuttling things around. Well, these NAD and FAD, or NAD plus and FAD, carry electrons around. So as these reactions break apart sugar, electrons are going to be released. And we need to catch those electrons because those electrons at the very end of this process are going to be so important for manufacturing ATP. So we're going to use these little electron shuttles to catch those releasing electrons and take them where they need to go. And so they can exist in the oxidized form of NAD plus or the reduced form where it gains some electrons. And you'll notice if this is going to be neutral, a positive ion and a positive ion is two pluses. We need two minuses to get it to a neutral one. Same thing here. If we're neutral and we're adding two pluses, we need two minuses to get it to be neutral. So everything has to balance. When you're looking at equations, remember, you can't make or destroy energy. You can't make or destroy matter. So everything when we look at these equations has to balance. And it may feel like you're kind of doing some accounting, like balancing a checkbook. But if everything's balanced, you're going to be in a good place. So when we look at something like NAD+, we get some of these hydrogen ions and some electrons. Attach that hydrogen ion and a couple electrons to balance out those energy levels, and we have a nice electron-reduced carrier to take things around. So, first step of the pathway. Every cell does it on the planet. Glycolysis. This is going to happen in the cytoplasm of the cells. So, first step isn't in the mitochondria. Now, why is that? Well, if every cell does it, not every cell on the planet has mitochondria. Prokaryotes like bacteria don't have organelles. Remember, a mitochondrion used to be its own prokaryote before it was swallowed up by eukaryotes. So because everything evolved from that ancestor that first did glycolysis, and that first ancestor had to have been a prokaryote because it was before organelles were introduced to cells, it would naturally take place in the cytoplasm because prokaryotes only have cytoplasm. They don't have organelles. So what is it going to use? It's going to use glucose, which has 6 carbon atoms in it. It's going to use 2 ATP. That we were making ATP. Well, we're going to get some out of it in return, but we got to put in some energy. Remember, reactions sometimes require those activation energies, and then 2 of those oxidized electron shuttles. We'll end up with 2 pyruvate molecules, which are 3 carbon molecules. So that means we're going to break apart sugar into 2 pieces. We're going to end up with 4 ATP for a net total of 2, since we spent 2 to get there, and 2 electron shuttles ready to go. So, as I said, 2 ATP are invested, but 4 are produced. So you're doubling how much you put in. So you can see as glucose, an ATP gets put in to modify it, another ATP to modify it, and then that is broken into 2 3-carbon molecules. In a process, electrons are released so we can reduce our NAD pluses, and there'll be some energy in that reduction that we can turn into ATP. Again, we'll have another reaction where that we can harness some energy for ATP, and we end up with a 3-carbon atom called pyruvate. And remember, since that was broken in half, we'll end up with this happening twice. So we'll get ATP once, twice, and then again, thrice for a fourth, 4 ATPs. So to look at it another way, 2 ATP go in, 4 come out, we end up with pyruvate and a whole lot more energy than we started with. And it's important to know that this is substrate level phosphorylation. So when we're adding these phosphate groups to ADP to make ATP, we are using enzymes to do this. So remember I said an enzymatically controlled reaction. Here's our first enzyme, attaching that phosphate group. Anytime a phosphate is added to something, the enzyme that does that is going to be called a kinase, which attach those P's to ADPs to make ATPs. A lot of letters being thrown around right now. But that's going to be substrate level phosphorylation compared to chemiosmosis that we see later. So we have aerobic cellular machinery in our bodies, things that are going to use oxygen to make lots and lots of energy. That's called a mitochondria. And they have double membranes that are absolutely necessary. So if we're going to breathe oxygen and use aerobic respiration, after glycolysis, we're going to do a link reaction to get us set up for the Krebs cycle, also known as the citric acid cycle, and then something called oxidative phosphorylation, which will take place in the membranes of the mitochondria. So let's take a look. But before we get into there, we have to do a link reaction. So pyruvate from glycolysis is going to enter into the mitochondria, and it's going to get modified a little bit. So we have 2 pyruvate molecules, so we're still at 6 carbons, 2 3-carbon molecules, and it's going to get converted to something called acetyl-CoA. Now you may remember your acetyl groups are carbons double bonded to an oxygen with another carbon there, and that's going to be attached to something called coenzyme A, which is another electron shuttle. It's going to be another coenzyme that's going to shuttle this acetyl group into the mitochondria. Now, out of this process, we're going to ditch a carbon, which we breathe out as CO2, and we get another NADH. Since we have 2 pyruvates, we're losing 2 carbon dioxide, so 2 of our 6 carbons are out the door, and we get 2 more NADHs. Cool.